Imagine placing foreign DNA in a bacterium. Will this DNA multiply? It will not, as the bacterium will not recognize the foreign DNA. To get recognized and to multiply, foreign DNA requires some identification sequences or replicon. These identification sequences are provided by a cloning vector. It is a DNA molecule which has an origin of replication and is capable of replicating in a bacterial cell. Most cloning vectors are genetically engineered plasmids or bacteriophages. Typically, the number of bacteriophages per cell is quite high. Therefore, the copy number of their genome in the bacterial cell is also quite high. On the other hand, the copy number of the plasmid genome varies from 1 or 2 copies to 15 to 100 copies per bacterial cell or more. When we link an alien piece of DNA with bacteriophage or plasmid DNA, we can multiply its number to be equal to the copy number of the plasmid or bacteriophage. The first artificial plasmid, which is now commonly used as a cloning vector, is PBR322. It was created in 1977 and was named after its Mexican creators, Bolivar and Rodriguez. Let us now review the main features that facilitate cloning into a vector such as PBR322. These features are origin of replication, selectable markers, and cloning sites. The origin of replication, or ORI, is a sequence of DNA at which replication gets initiated in a plasmid. When a DNA piece is linked to this sequence, it can be made to replicate within the host cells. This sequence also controls the copy number of the linked DNA. Therefore, whenever one has to recover many copies of the target DNA, it should be cloned in a vector whose origin supports a high copy number. The vector also requires a selectable marker, due to which a plasmid is more useful for the cell. Selectable markers help identify and eliminate non-transformants and exclusively permit the growth of transformants. For example, in E. coli, the genes encoding resistance to various antibiotics such as ampicillin, chloramphenicol, tetracycline, or kanamycin are used as selectable markers. Another important feature of a cloning vector is a cloning site, which is an artificially constructed region within a vector molecule containing a number of closely spaced recognition sequences. These sequences or recognition sites can recognize several restriction enzymes, and therefore the vector can be used to clone a variety of DNA samples. However, the presence of more than one recognition site may lead to complications in gene cloning. In the cloning vector PBR322, the ligation of alien DNA is also carried out at a restriction site present in any one of the 2 antibiotic resistance genes, which are ampicillin and tetracycline. Let's take the example of foreign DNA which can be ligated at the BAM-H1 site of the tetracycline resistance gene. When foreign DNA is ligated at the BAMH1 site, it disrupts the tetracycline gene and consequently the recombinant plasmids will lose tetracycline resistance. Then, the transformants growing on the ampicillin-containing medium are transferred to a tetracycline-containing medium. In the tetracycline medium, there is no growth of transformants. Meanwhile, non-recombinants grow on the medium containing both the antibiotics. Therefore, while one antibiotic resistance gene helps select the transformants, the other one gets inactivated due to insertion, thereby helping in the selection of recombinants. The selection of recombinants due to inactivation is a difficult procedure. Therefore, different selectable markers have been developed which can differentiate between recombinant and non-recombinants by producing a color in the presence of a chromogenic substrate. For example, when a fragment of DNA is inserted in the middle of the coding sequence of an enzyme beta-galactosidase, it results in the inactivation of a beta-galactosidase gene. Due to the addition of extra codes, any future products from the inactivated gene will not work. This is known as insertional inactivation. When the plasmid that has undergone insertional activation comes in contact with a chromogenic substrate, colorless bacterial colonies can be observed. These colonies are also identified as recombinant colonies. On the other hand, if the plasmid in the bacterium does not have an insert of foreign DNA fragment, the chromogenic substrate yields blue colonies. Interestingly, human beings have learnt about cloning vectors and the concept of gene transfer into plants and animals from bacteria and viruses. Consider the case of Agrobacterium tumefaciens, a pathogen of several dicot plants which causes tumors in them. It delivers a piece of DNA known as tDNA and transforms normal plant cells into tumor cells. These tumor cells are used by the pathogen to produce chemicals such as nopalin required by it. By understanding how pathogens deliver genes in their hosts, scientists have been able to transform the pathogen into useful cloning vectors. For instance, the tumor-inducing Agrobacterium tumefaciens has been modified into a cloning vector Ti plasmid, which is not pathogenic to plants. In fact, the Ti plasmid vector uses the same mechanism to deliver other genes of interest in plants. Once we have obtained the recombinant DNA molecule using a cloning vector, another challenge is introducing this recombinant DNA in the host cells. In DNA, the deoxyribose sugar and the phosphates are hydrophilic in nature, therefore cannot pass through the cell membranes. To overcome this problem, bacterial cells have to be made competent to take up plasmids. Therefore, they are treated with divalent cation like calcium. This enhances the efficiency of the entry of DNA into a bacterium through pores on the cell wall. After this, Recombinant DNA can be forced into such cells by incubation of cells with recombinant DNA on ice. This process is followed by administering a heat shock to the cells at around 42 degrees Celsius and putting them back on ice. After this, the bacteria are able to take up the recombinant DNA. Apart from cloning vectors, foreign DNA fragments can also be introduced into host cells using methods such as microinjection and biolistics or a gene gun. The microinjection method involves the direct injection of recombinant DNA into the nucleus of an animal cell. While in the biolistics or gene gun method, plant cells are bombarded with high-velocity microparticles of gold or tungsten coated with DNA. This method uses disarmed pathogen vectors which, when allowed to infect the cell, transfer the recombinant DNA into the host. The use of cloning vectors and methods such as microinjection and biolistics are crucial in genetic engineering as they help create genes of human and scientific interest.